Hello, good evening, everyone. Hope all is going well at your side. Sorry, I was for a long cycling today in the evening. I went around 6:30, and this in between there was a lot of traffic, and I could come by 8:30, but I didn't want to make it fast because of the traffic, and it was quite dark. So I made it to 9 p.m. to come to my room, and we can start this class safely. Uh, So let's start with the basics bioinformatics of basics information about bioinformatics, an overview about bioinformatics today. We're gonna initiate our class with that. So just wait for 1 more minute and then we start. Okay. So basically, bioinformatics is a mixture of all the techniques. It is also like we were discussing our first day with the interdisciplinary nature of molecular biology and biochemistry that are being used in many places. The same way, bioinformatics is also playing an essential role, quite an essential role in making things working for all of us. Yeah. So some biological concepts, a brief history of a young field, and where data is coming from, and large scale biology. Some new words, new techniques, like all the omics techniques we will discuss here. So basically, as I discussed, bioinformatics is an interdisciplinary field which is having biology, mathematics, statistics, informatics, biophysics, all are coming within this field. So basically, some students who are doing IT in their bachelor's, information technology, they opt for bioinformatics as their masters. So one of my colleague, the Manjeet Kaur, she's from Sweden, and they took I mean, she when she was doing her masters in bioinformatics, most of the students that she was having there, they were all from the information IT background, because this bioinformatics field was so much coming up in upcoming time. So all the, like, now the vaccine came now, so in order to find out the structure, it's very hard to find in the wet lab and to work around it. So bioinformaticians come and they do structural information about it and get to know about it in a very, you know in a quick manner. So the work that has to be done in 2 or 3 years, they will do it within 2 weeks. So that's how important the bioinformaticians are. So in each lab, in each group nowadays, one bioinformatician or a small group of bioinformatics people are really essential. And beyond that, within this bioinformatics field, it's also very easy to grow up, because there's not so much competition at the moment, but it will be in the future, because not so many students are doing this. Everyone is going on the wet lab side. But so whosoever is doing this bioinformatics, I'm pretty sure with the languages, Python, R language, and various other languages. So those students with the IT plus biology, they will be the most required people in upcoming time. At the moment, it's higher salary they are being paid, and but yeah, it's a sitting job, and you predict many things, and you got also, money wise is also very fancy field. So as we know, this DNA, according to the central dogma, it's giving RNA, RNA is giving protein. So study of DNA is giving us a DNA genome database with a cDNA and transcriptome. from RNA, and the protein is giving us protein sequence and structure database. So we are getting out of protein then a proteome and a proper phenotype. So according to NCBI, the main definitions came out to be of bioinformatics is a field of science in which biology, computer science and IT, they merge into single discipline. So all the biology that we have studied, the basics, and the computer science. I don't know, for myself, as me as a person, whenever I started to learn about C plus in my plus one, when I was asked that you want to take computer science, I was very afraid. I got like goosebumps in the night. Somebody told to write this, um, to write one language, one program of plus and minus. Till that time I was very happy, but later on it's got complex. Some yes or no, not some more data, more languages were coming up, and I was like, no, I don't want to go for it. It's not for me. So find your own niche. That what is your niche is what you're good in that. Then go and find it, because it should be something that it's called Ikagai. Recently I made one video. I hope you have seen that video. It's sorry to go beyond this topic, because I really want you all students to think, because some students I had messaged that they want to go for food biotechnology, or somebody was asking to go abroad, somebody was asking to do some other aspect of field. Yeah, so everyone has their own goal, has their own will. And so this is the Ikagai. It's good to discuss some general stuff also beyond our daily studies. That means that you have to find a reason for being. That if somebody asks you that you have to work from Monday to Saturday, or Monday to Friday, so you have 2 days off, and by the Monday you have to work again. That you should not be like, oh, Monday is still there, I have to go to work. It should be, oh yes, Monday is there, I can work more, I can do many things. If somebody is software engineer, he loves doing coding. He can't live without coding. His hands, his brain, it's just meant to be do coding actually. And that person is doing marvelous stuff. That person who doesn't have an interest in that. So it's just like, I will ask you all students. At the moment, maybe few students have joined me. Later, I think some students joined, 13 students. So you all 13 or 12 students here, just think like what you really like to do it, which makes you happy. Like while doing it, you have a flow while doing it. You know what is flow? Like if you started to read a book, you want to keep reading that book for whole night. This book had made me to read like that. So I just got this book or online order, and one day came, I started to read, and within one night I finished it. And I made just one YouTube video about also, because that books really made me strike that why I'm here, what is the reason on being earth. The same thing, you all have to discover it. You might have discovered it already, or soon you will discover it in upcoming time. The day you will discover it, you will get to know why I have to wake up out of my bed in the morning, the reason for getting out of the bed. Yeah, for a mother it's different, for a father it's different, for a like daughter it's different, for son it's different. Everyone has their own role and their own being, what they want to do it. So you just have to discover those things. So that is Ikagai. Ikagai is a Japanese concept meaning reason for being, which lets you get off the body out of the bed. Yeah, so you can watch that video. It's available on YouTube. Just search Ikagai. It's a 10 minutes video. So yeah, same here, that when you want to do it, just think, if you are very interested with bioinformatics, then I would definitely go for it, because you get to know about statistics, new algorithms. Like if me as a molecular biologist, I started to analyze data. So for me, if I started to do it, so it will took me around 2 weeks or 1 week to analyze the data. But these bioinformatician people, they create some formula with machine learning and artificial intelligence. They will create one coding formula. You just you have to just add some figures. You will automatically, your data will be analyzed automatically. So that's the beauty of it. Yeah, so there are many at the moment workshops are being going on with like machine learning workshop in biotechnology, or machine learning in biology, or there's also many courses available. So I will highly recommend to go for it. There are three domains of life, tree of life, that is archaea, bacteria, and eukarya. According to that, we have divided our subspecies, chloroflexi, bacterotides, and so on. Yeah. So bacteria, archaea, this is candidate phyla, Dapanan, Locriacota, and Eukarya, and so on. So this is the like various difference, within the field of this, and there are two cell types, that is bacterial and archaeal cells, right? So bacteria and archaeal cells, bacterial cells includes your nucleoids, cytoplasm, cell wall, and cytoplasmic inheritance, submembrane, sorry. Whereas the eukaryotic cells, it has more intense structures as compared to that, and the central dogma of both a bit different because of that also. So in this, we having like in the prokaryotes, gene A and B, after the transcription, according to the coding region on the mRNA, they will produce coding region A and B, and then protein A and B. Whereas in the eukaryotes, it will has intron and exons within the nucleus over the gene X, and then they will produce out the transcription exon 1, exon 2, and exon 3, and also introns itself with the primary RNA transcript. Then mature RNA, it will splice these introns, they will be removed. At the end, you will have mainly exons, and they will be transported to the cytoplasm, and then hence with the help of translation, you will have your protein within that. So that's how this is taken care. So protein splicing, yeah, this is a mature RNA, introns are removed in that case. So in bioinformatics is generally, just a second, Siren. Oh. It's about genetically coded information and protein. So if we're having these three TT, GTT, this double helix structure of DNA, it could also replicate producing this transcription UU, GUU, AU, and then the translation with the various amino acids, phenylalanine, valine, aspartic acid, glycine, histidine, leucine, and so on. So this is how DNA, RNA, and proteins are being made. So it's about, so how we have to see, we have seen these data within our body, and we get to know about with the help of protein sequencing, with the that we did this Sanger sequencing, next generation sequencing. We get to know about all these data, but it's very difficult to analyze at once. So in that case also bioinformatics is playing a big role. So it's all about data management, database structure in which we analyze and interpret the data using the web-based or standalone tools like FASTA, BLAST. I hope you have done those common things in your bioinformatics classes. And then there's a writing code, Perl, Python, and Java. So these computer languages, and further to develop new algorithms and statistics for the analyzing the data. So that's basically your bioinformatics is playing a role there. And as we know, within this bioinformatics, also for the genome, we have this in 2003, I guess. This whole genome was discovered, 2003 or in some other year maybe. It was very difficult to find out whole genome of human body, but all the 23 chromosomes and their respective, DNA sequence was discovered. From that day, the whole thing started to change. Bioinformatics started to play a big role. So in summary, bioinformatics help us to uncover biological information hidden in the mass of sequence, structure, literature, and other biological data, and train on insight into the fundamentals biology of cell organisms. So genomes give us transcriptome, from that we get proteome, and then metabolome, and then the phenotype out of it. So here we can see there is a computational tools and infrastructure on the y-axis. and on the x-axis the biological complexity. So according to the complex, as much as your things are getting complexes, the various tools are being used at that point. So if we see here, the simplest way, first you study the comparative genomics, in which you compare the genomics of various species. Let's say human with the mice, or tapeworm, or dragonfly, or any other species. You check each chromosomes one by one, each genes, that which genes are common in each other. Then comes also docking, rigid docking system. Rigid docking means you check the substrate and enzyme whether they are getting fixed or not together. Then cell pathways and network stimulations, to know about the cell signaling pathways within the cell. Then molecular machine classic simulations is also being discovered with the help of that. And then molecular based cell simulation, that we simulate the cell and then we check the how molecules are behaving within that. So this is all hypothetical based bioinformatics. So there is also fight between bioinformaticians and the persons who are doing wet lab or molecular biologists. So they are saying the people who are working in the lab day and night, and they say that we are doing and we are finding the real results, that what is happening in the cells. But these bioinformatics results, they are all prediction. They are not the true results. They are all based on predictions made by the software in the computer. So that's something thin line going in between. So there's a raw data sequence is also possible, like we having these 4 bases A, G, C, T. So we can get to know what which are coding and non-coding. So if you want to discover some particular sequence within the whole genome, so with the help of bioinformatics like ATG or GCCG, AAGGC, we have to discover these sequences. We can find out where it is lying within the sequence of your DNA. And beyond that, section enzymes are quite sequence specific, as we have discussed in the day 4 class. So they recognize and bind to specific DNA sequence. An enzyme E. coli, for example, binds to 6 base pairs sequence to GATTCC. So they, as we know, they give this palindromic sequences cutting from here and here. And then how often this, and we have to discover that if E. coli is cutting in this sequence, from which part it will be cutting in a random DNA sequence. We can get to know what from these parts. GAATTC. Yeah. So this is also possible. This is some basics. We know that purines and pyrimidines, adenine and guanine. Pyrimidine includes thymine and cytosine, whereas the A with the T pairs 2 hydrogen bonds, G with the C3, and genetic codes, they are into like 4 into 4 into 4, like 4 is to power 3. That is 64 total triplets of nucleotides are present, and that's how the protein sequence is is being distributed over the UnitProt, SwissProt sequence database. So we have majorly your bacterias. Yeah, bacterias are highly present, like around 16%, and then comes your archibacteria. Then comes your little bit of a eukaryote and viruses. So the protein sequence that we have at the moment is highly because of the your bacterias, and then the eukaryote, then the archie, and then the bacteria. So in the protein database bank, if you go, PDB data is there, protein database. So you can get to know about the protein structure, that how your protein structures looks like. This is the PDB molecule of the month, some tRNA. This is your tRNA with the AAG, CUG, aspartate tRNA, and phenylalanine tRNA present here. So you can find out various structures about various proteins on the PDB website. Now some historical landmarks within this field of bioinformatics from 53. First, your DNA was revealed, like in 1953 by Watson and Crick. Then in 55, your protein sequence insulin was discovered by Sanger. Then first enzyme by ribonuclease in 60s, and also the genetic code were elicited at that time. And 77 again DNA sequence by Sanger and Gilbert method. And 75 first human gene was isolated. Then 82 gene bank database is set up. Then PCR was first discovered in 83 by Carey Mullis. Then fluorescence based DNA sequence in 86. The term genomics, the first in 86 was coined. Then 91 bioinformatics appeared in the scientific publications. somehow people get to know what about 1991, that what bioinformatics is all about. In 1995, complete genome sequence of free living organisms. In 1999, human chromosomes 20 were sequenced. In 2001, first draft was ready for human genome. And 2003, yes, I was correct, first human genome project was completed. And since then, we never see back, we tend to go up and up and up and up. So that's how the bioinformatics, if you put over PubMed, computational biology, computational molecular biology, molecular bioinformatics, biocomputing, all these are corresponding fields to bioinformatics. And PubMed search with related the word bioinformatics, it tends to increase by the 2003 since the your genome was discovered. Then since then it's just keep on increasing, keeps on increasing, results by year. So main historical landmarks that happened are in 1970, Needleman Wunsch, who discovered the algorithm, and Chow Fastman, they predict the protein secondary structure for amino acid sequence. And in 81, Smith Waterman algorithm for sequence alignment. In 82, Kite and Doolittle hydropath plots. In 88, Pearson and Lipman fast heuristic similarity. And 87, progressive sequence alignment. Then 1990, basic local assignment search tool, BLAST, yeah, and SciPLAST in 1997 were discovered. So after the knowing the DNA sequence, there was exponential growth in other sequences also, not only the our human, but also other sequences were discovered. So National Library of Medicine, 24 years of growth. It has also discovered many other database such as PubMed was discovered 1997, your human genome in around 1999, then NIH Public Access in 2005, Genome Wide Association Studies GWS in 2006, and genome testing in 2012, MedGen Reader in 2013. So all these, Blasta in 1990, Enters in 1991. So all these discoveries, the key discoveries that has been discovered, they have been very exponential role in finding the things. You can see users, that the people who are using, initially there were not so many people who were using, but since the discovery of a human genome, it was abruptly started to increase. So the users increases, the gene base pairs is also increased. So everything started to increase since 2000 year. And then this DNA sequence with chain terminating inhibitors by the Sanger, which was published in PNS paper in 1977, and for the same, Sanger got the Nobel Prize winner. So high throughput sequencing technologies, next generation sequencing methods, AGCTAG. So in 1997, 77 first sequence organism, bacteriophage, was discovered. Yeah. In 1995, first living organism like Haemophilus influenza was discovered. And then 1996, first eukaryotic sequence was Saccharomyces cerevisiae. So this used to come in objectives MCQ sometimes to make you confuse. So which organism, first living organism, yeah, first eukaryotic organism. So just remember these different names: bacteriophage, Haemophilus influenza, and Saccharomyces cerevisiae. Yeah. Then some sequences capabilities versus computational power. So we can see bases per day that the sequences, it starts to also keep increasing, and the computational power was tend to bit less than as per the sequences start to come. So sequence capabilities are doubling approximately every 4 months, whereas the processing capabilities are doubling approximately every 18 months. So it was tend to go slow because your data was generating huge amount, but to process that data, it needs some time. No, it's not easily possible. So this is one another thing, the other sequences that were discovered later on. So in the series wise, like Homo sapiens, draft was created, then Melongster, C. elegans, cerevisiae, tuberculosis, human influenza, Fred Sanger method, HIV genome sequence. So all were discovered one by one, and keep going on. So this is the in 14th April 2003. This was published in Nature. One in 1999, the chromosome number 22 and chromosome number 21. Their sequence were discovered one by one, and then by the end in 2003, this is the first the draft of human genome sequence. These 24 chromosomes, 22 are allosomes, and these rest two are your sex chromosomes. So Y Y Y X X X X X X. then 22 these allosomes. So you can open these books, you can check these sequences, so all the sequence will be merged in them. But this is very difficult for people to read them and to use them for their data. So that's why these bioinformaticians, they play a big role. There's all these database now available on internet, and we can analyze, we can mutate, we can play around it to create transgenic, to create mutation, to create knock-in mouse, to create knockout mouse, whatever you want to play around with the genes, with the cloning, you can freely do that now. So now new methods, new words. Genomics refers to the analysis of complete DNA sequences, and functional genomics, the study of obtaining an overall picture of genome. Whereas the proteomics, it's a protein expression of a genome, which refers to as study of whole proteome. Like we have discovered this 2D gel electrophoresis. So from that we get to know about proteome, about full proteome, and we can study about all the protein parts. Then transcriptome, it's a complement of mRNA transcribed from cell genome, and transcriptomics is the generation studies of the complete mRNA expression profiles. So structural genomics and proteomics, so assignment of it's a three-dimensional structures. To get to know what it is sequence of hypothetical protein, peroxidoxin. So you can play around with the sequences also. Then comes omics. Yeah, this is also an integrative biology, a new branch which is playing very big role, like genomics, transcriptomics, proteomics, metabolomics, pharmacogenomics, physiomics, and so on. So genomics, it's basically the study of genes, regulatory and non-coding sequences, and transcriptomics is RNA and gene expression, and proteomics is the protein expression, whereas the metabolomics is the metabolites and metabolic networks. And pharmacogenomics is the quantitative study of how genetics affects host response to drugs. And physiomics, physiological dynamics and functions of whole organisms. This is transcriptomics. In this, if you have a cell or tissue sample, you get the cellular RNA out of it, tRNA, mRNA, sRNA, and then you do the microarray approach, or you do the RNA sequence approach. In the microarray approach, you label them with the complementary DNA and RNA, probe them, and infer presence of transcripts, analog the quantification. And on the other hand, in the RNA sequence, you compare the target sequence that you are looking for. Then microarray approach. So yeah, so we have a bacteria grown in culture, label complementary DNA from bacteria with a red Cy3D, and on the other side is the bacteria from site of infection, label cDNA from bacteria with green C5D, Cy5D dye. And then you co-hybridize them and put them over the DNA microarray. Then you get these yellow, green, and red dots. So green one the express the infection only, red not expressed, the blank one. Yellow are the both culture and infection, and the red are the expressed in the culture only. So RNA sequence, in this we have to isolate mRNA, convert it to the complementary DNAs using transcriptase and sequence the DNA, then map sequence to the genome, so your mRNA is fragmented, and then the reverse transcription is done to analyze or to have cDNA, then we sequence and then quantify it. So the more times a given sequence is detected, the more evidently it could be transcribed also. So how proteomics is working, basically you have a sample preparation, you have a proteome, then you digest the sample with the trypsin, then you can separate them with the HPLC, high performance liquid chromatography, and then you put your samples into the mass spectrometry. Yeah, so where with the ionization your protein part, they got separated from each other, and then you have ions which can have this MS full spectra. So we can see various ions which belongs to which part, like insulin and beta lactoglobulin we have discussed in that protein isolations end part, and then you can quantify it, like how much which present and which thing, and then you can go MS precursor selection, afterwards fragmentation is done, so you have a double spectra MS, and then you can identify further to have more details about your protein. So there is a protein diversity also. We can also find out that like various post-translational modifications happening, like glycosylation, redox modifications, ethylation, methylation, and many more. Then it's also protein bioinformatics is essential in the system biology in order to study the planet. Yeah, we have studying planets with the ecosystem, and then the community and organisms and various ecosystems like system, organs, cell, and DNA, and so on. This is a software not required. So top 10 challenges that we are seeing in the as a bioinformaticians is to precise models where and when transcription will occur in genome, precise predictive models of alternative RNA splicing, precise models of single transduction pathway, determining the protein like is it DNA, protein DNA or protein RNA or protein recognition codes. Further, rational design of small molecules inhibitors of proteins, mechanistic understanding of proteins, yeah, mechanistic understanding of speciation, further education development of various bioinformatics curriculum. So that's other at the moment the 10 challenges. So that's it from the bioinformatics perspective. Uh, um, quiz is will be MCQs questions. There will be 30 questions there, and there you have to answer them in within 40 minutes. Yeah, you will be given 30 questions and you have to answer them for 40 within 40 minutes. Each question will carry 1 marks, and there will be no negative marking. So the top 5 students will get the appreciation certificate out of all the students who will give the exam. And yeah, last time they were like around 8 students. They were like tie was happened. So somebody got 29, 27, 27, 28, 28. So that's how we distributed the things. So hope for the best. So that will be done after the classes are over. So I will take that thing over one day. So maybe it will happen after the today we are done with the bioinformatics part. So one day we will do with the I have to do this X-ray crystallography also. Our X-ray crystallography is left. So I will play that topic now, and then yeah, on Friday we will have the test. I will write in the group. I will write in the group. So basics of protein crystallography and X-ray diffraction. So here you will be acquainted with the basic principle of X-ray, phenomena of diffraction, how this could be deciphered in the structure of protein, and you will get the in-depth knowledge of instrumentations, mechanistic overview of X-ray crystallography. So why structure determination? Why we have to do this structure determination? It will help us to know the protein interaction with other molecules, how they perform catalysis in the case of enzymes, and interaction of proteins with other molecules including your protein itself. So miscoding or misfolding of proteins associated with the disease. So this is all possible with the field of X-ray crystallography. So what is X-ray crystallography? It's basically a high resolution microscopy which enable us to visualize protein structures at atomic level and enhance our understanding of protein function. So here we have seen that we have a crystal structure. You put on an X-ray light on it, and then it will be diffracted over this detector. So based on that, we can get to know about we can predict the protein structure, and that's almost accurately find out. So in microscopy, so the microscopy, the wavelength is around 300 nanometer, and we can get to know about the individual cells or subcellular organisms. And with electron microscopy, we can get to know about 10 nanometer of wavelength to deep structure. We can still see very deep structures. But with X-ray, which is having a wavelength of 1 angstrom, we can get to know about the atomic detail of proteins actually. So which protein, which amino acids, where is it acting, which kind of double bonds they are making, what kind of hydrogen bonding they are making, we can get to know about that. So basically in X-ray is the order of atom is in the order of atom diameter and Bohr lengths, and allows oopsie doopsie. Yeah, allowing these to individual resolve, and low lenses available to focus X-rays, so crystals act as a magnifier in this case. So what is the main steps that are taking place in this is first protein purification, then protein crystallization, then data collection, then structure solution, and then structure determination. Yeah, first you have a crystal. Yeah, you put over the X-ray, then you make it over the diffraction pattern, and then over the faces you have electron density maps, and then you fit over the atomic model. Then your refinement is done of that. That's the basic way of doing the X-ray crystallography. So in the first is a protein purification that we have discussed in previous chapters, that in the series of process intended to isolate one protein from your whole tissue, and then we characterize that protein to know about its function, structure, and with which other proteins it is interacting. So we need around 5 to 10 milligram per soluble, and it's better to have 95% pure at the end. Then we have to crystallize that protein. So like if this one molecule is there, so this is the unit cell. So your crystal will be like all these molecules coming all together within this one box. So in this X-ray crystallography, your form a single unit would be very unmeasurable weak. We can't see from the single unit. So we need to create a crystal to see our results. So protein crystallization, crystal must be small in size, less than 1 millimeter, no cracks, no incursions such as air or bubble, right? So hanging drop method is there. So 1 to 5 microliter protein solution is suspended over the 1 ml reservoir containing the precipitant solution. Example, ammonia sulfate solution or polyethylene glycol, and so on. So in step 2, afterwards, once you have your crystal ready, you put them over the X-ray light. So this will do the Bragg's law, in which they will get deflected the straight forward, and then you will get your results on the detector. Then this mounting is crystal, like you have to put your crystals over this sample. So crystals are mounted in a way that samples can be rotated and X-ray beam can be passed through the sample on a 360-degree value. From all way it could be give this value to it. And methods of mounting including using either a capillary or tube, and both capillary or tubes are mounted on the genometer. And there is a synchrotron, then once the crystal is mounted over there, then with the X-ray beam with the synchrotron, which give a high resolution luminosity of X-rays, or X-ray generator we can use for smaller laboratory use. Then with the help of source of X-ray is often synchrotron, and crystal data collection is done on these dimensions, and then we get these data. So you have your rotating anode here, and you put electron on top, and then X-ray beam are being given there over the prism, and they are overgone into the crystal, and then this is your chi circle, and then you're getting the results on the area detector. So at the end, data collection is done. So this is the kind of data that you see. You see, dot 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 dot. So like say if we having a car, and you are having a car, and you hit with the ball. Yeah, you hit with the ball, and ball will hit and will go up there. You hit the ball from here and go up there. Hit ball there and there. So we can predict that by by hitting it, where it is going, how much angle it's making it. So we can make one prediction. So might be this place might be a bit higher than this. This might be a bit slant than this, and this place might be flat than this. So we can predict many things according to the as the X-ray rays has been being putting up over the protein structure. So by this we can having this prediction structure, like this might be the structure of the protein, and this is almost right actually. So in overall we can get to know about this basic operating principle of X-ray and its application proteomics. You are able to understand the principle of proteins and how the data is elucidated afterwards, right? So that was something quick about X-ray crystallography. So now let's check one Bragg's law, how the Bragg's law works in it. This is also famous because they got the Nobel Prize winner for the same. Now, one last video. Okay, a very smart scientist explaining about basics about the extracrystallography. So I took you a lot of time. You might be sleepy. It's time to go bed. Yeah, have a nice sleep. We see us tomorrow on the right time, actually, not in the night. So wish you all a nice evening. Take care. I will send these datas after the class is over. Yeah. Good night. Bye bye.